Hello friends, good afternoon and welcome to EduSat Live Lectures. Dear friends, today in developmental biology we will be talking about regeneration. To discuss this topic we have with us our subject expert Dr. Charu Dogra Rawat. Dr. Rawat is Assistant Professor in Department of Zoology in Ramjas College, University of Delhi. Without further ado, I would like to welcome ma'am to our studios and request her to start the lecture. Welcome ma'am.
So, apart from metamorphosis, another strategy for post-embryonic development which is found in some of the animals is regeneration. So, as the name suggests, regeneration basically means that generating the parts again. So, it's basically the developmental process is reactivated after the baby is born or after the birth of the animal is there and it has grown up. So, just like metamorphosis, this is again post-embryonic development. So, the learning objectives for this session would be to understand the three regenerative modes which are the epimorphosis, the morpholexis and the compensatory regeneration, and we will take one example, at least one example of each one of them. To understand the mechanism of regeneration in salamander, newt, to understand the factors affecting these regeneration in salamander, and to understand the mechanism of regeneration in Hydra. So, basically salamander and Hydra would be the examples for epimorphosis and morpholexis, and we will be taking and learning the mechanism of regeneration in these organisms. So, what exactly is regeneration? Regeneration is the reactivation of development in post-embryonic life to restore the missing tissues or the lost part. It involves the systems in which removing a complex structure or major part of an organism initiates a chain of events that produce a structure that duplicates the missing part both functionally and anatomically. So, functionally and anatomically is very important because the regenerated part is equivalent to what part was already present in the organism. So, basically when a tissue is lost or when there is an injury, then instead of a repair, generally we have a repair mechanism that whenever there is an injury, the mechanisms or the signaling events will start and that wound will be repaired. But usually in mammals, for example, if you may recall, then there is a scar formation which is there. Regeneration involves that the entire part or entire wound is basically treated in a manner that the part which is then developed is identical functionally and anatomically to the original part. And for this to occur, the reactivation of the entire developmental process takes place. There are three different regenerative modes. One is called as the morpholexis, in which the basis of regeneration is differentiation. There is a repatterning of the existing tissues with very little new growth. So, we will be discussing the examples of all these three different types, but you have to understand the difference between the three regenerative modes. So, in morpholexis, which is exhibited by Hydra, a coelenterate, the repatterning of existing tissue occurs with very little new growth. However, in epimorphosis, there is a de-differentiation which occurs. It is exhibited by the urodale amphibians such as salamanders, and the adult structures de-differentiate to form a mass of cells that re-specify. So, that is basically the epimorphosis. In some organisms, for example in mammals, we will see there is something which is called as compensatory regeneration, for example in mammalian liver. In compensatory regeneration, there is a proliferation of cells, there is a growth of cells, and then there is a differentiation that occurs. So, the cells divide, but they do maintain their differentiation functions. So, you know that when the cells are growing or when during the developmental process, it all starts from a single zygote, and then there is a mitotical divisions, the cleavage that occurs, then there is gastrulation that occurs, which is the specification of the three germinal layers, then each germinal layer give rise to certain organs and the adult structure is formed. And when there is an internal fertilization, then the baby is born, or the baby is born and then it grows to form the adult. These post-embryonic developmental process almost reinitiate the developmental processes which were taking place while the organism was formed. So, all these mechanisms involve one or the other thing, the proliferation and the redifferentiation or dedifferentiation as it is called as, or the differentiation, retainment of the differentiated functions are also there. We can understand this two modes of regeneration, the morpholexis and epimorphosis, by taking this example of an intact French flag system. So, for example, there is a positional value for each. So, there is a French flag which is shown up, and the cross basically refers to where the cut occurs. So, if the left half is removed, that means the tissue is lost and only the right part remains. Then in the case of epimorphosis, there is a growth that occurs, and due to that growth, the cells will divide and will again differentiate to form the rest of the part of the flag. So, in epimorphosis, the grown part is exactly identical to the original part, and it is of the same size. You cannot distinguish between the initial, original and the regrown or the regenerated part. But what happens in the morpholexis is that the tissue which is remaining, it basically gives rise to a new positional value, and as you can see, there is a rearrangement of the cells, or there is a redifferentiation of the cells that occurs, and that kind, there is not much growth in that, and there is a repatterning is the right word. So, repatterning of the tissue occurs and the entire flag is reorganized, but it is smaller than the original one which is there. So, this clearly depicts the difference between the morpholexis as well as the epimorphosis, and now we will take the example. Before talking about that, we will talk about a great scientist whose name is Thomas Hunt Morgan. You must have heard of him. He won the Nobel Prize in genetics, developmental biologist. He began his research career studying regeneration. He coined the term morpholexis and epimorphosis to describe the two major types of regeneration. And as we said, the morpholexis refers to the type of regeneration that occurs in the absence of cellular proliferation. There is no growth that occurs, while epimorphosis refers to regeneration that requires active cellular proliferation which is there. It has been reported that Morgan once said that since he had been unable to solve the problem of regeneration, he had decided to try something easier, such as the problem of heredity. So, his name is associated, Morgan's name is associated with heredity and genetics, everybody knows it. So, but he started working on regeneration, but it was not that easy to, uncover the mechanisms of regeneration. So, he sometimes, he once mentioned that is why he shifted on to the problem of heredity which is there. Then there is another kind of regeneration which is usually the stem cell mediated regeneration. It involves the use of stem cells to regrow the organs or tissues that have been lost. Its examples include the regrowth of, for example, hair shafts from the follicular stem cells in the hair bulge, and the continual replacement of the blood cells from the hematopoietic stem cells in the bone marrow in case of mammals. But we will not take up this topic right now, and we will discuss it when we discuss about the developmental genetics. So, now to start with the examples of the two modes of regeneration, which is the epimorphosis and morpholexis, the nature's paradigm for tissue regeneration is represented by this organism which is called as the salamander. So, it can practically grow back the spinal cord, it can practically grow back the tail, the heart, the retina and the lens, the limbs, the upper and lower jaw. So, it's a paradigm for tissue regeneration. It can probably regenerate many of the tissues of its body. And in the insect, it is shown that there is a lost arm which is there, and then it has been regenerated. In the slide, the diagram is already shown there. So, there is a cut part which is there, and then the regrown part is there, which is, so we will study the mechanism of this, how does this occur. Spallanzani was the scientist in 1768. He first reported that the Urodale amphibians were able to regenerate their limbs after amputation, and this was the series which was followed, or the stages of regeneration. We will elaborate it down the lane, but just to start with how Spallanzani explained it, there is an amputation, there occurs what is called as wound healing, then there is a dedifferentiation that occurs, and then after dedifferentiation there is an early bud that develops, then medium bud, late bud, then the pellet formation is there, early differentiation occurs, and the complete arm is regenerated. Regenerating a limb, a newt can regenerate an entire limb within 7 to 10 weeks. So you can see the growth cycle. 1 week there is a stump which is there. In 3 to 6 weeks a pellet and a cone shaped has been formed, and in 6 to 9 weeks the entire limb has been regenerated. So let's look at the stages of regeneration. There is an amputation that occurred, and it's the histologically also it is shown, and then there is a healing which is occurring. So there is a wound epithelium which is formed on the top. There is a dedifferentiation, this kind of an apical cap underlying this area. When there is a dedifferentiation of cells occur, it is called as blastema. So those blue are the blastema cells, and the upper yellow ones are the apical epidermal cap. Then comes the cone stage, in which there is a bud formation and it just elongates a bit. Then it flattens out to form the pellet stage, and then there is a knot stage, and then there is a digit stage when actual digits are laid and the entire limb is regenerated. Let's look a little deeper into all these. So the first thing that occurs is the wound healing. So within 24 hours of amputation, a layer of epidermis migrates across the limb stump, the wound epidermis closing the wound. This single layered structure then proliferates to form what is called as the apical ectodermal cap, or AEC. This is a critical step for limb regeneration. So in anywhere, whenever there is an injury, there is a repair mechanism that occurs, and the first part of the repair mechanism is the wound healing. So the same thing happens here. There is a wound healing, there is a layer of epidermis that grows up and covers the wound, and this layer is called as the apical ectodermal cap. Then occurs a process which is called as dedifferentiation. After the wound is closed, the cells of the stump undergo dedifferentiation. This involves a loss of specialized histological characteristics and a return to a less specialized cellular structure. That is, the dedifferentiated cells resemble the embryonic mesenchymal cells. For example, muscle cells lose their contractile apparatus and return to the motonucleate condition. Dedifferentiation occurs for some distance proximal to the amputation level. So this is basically why we say it's a reactivation of the developmental process. And in this particular example, what happens is that the cells lose their differentiated functions and they come back to their original form, their embryonic form. So they lose their specialized function, they become embryonic in nature, and then they re-differentiate to form something, give rise to the structure which it needs to. So this dedifferentiation is a very important process in case of regeneration of the limb of a newt. So you can see there is an amputation, the wound healing has occurred. So this yellow cells are the apical ectodermal cap, and then the dedifferentiation has started, giving rise to what is called as the blastema which is there. So the next is the blastema formation. The dedifferentiated cells then begin to accumulate at the end of the stump beneath the epidermis to form what is called as the blastema. In the blastema, mitosis occurs rapidly, and the combination of migration of dedifferentiated cells and the proliferation of these cells results in a large mass of morphologically undifferentiated cells. So this large mass of morphologically undifferentiated cells is termed as blastema, which is formed at the site of the injury. This mass of cells then begins to elongate to form a cone-like structure, and later the cone flattens to form what is called as a pellet-like structure that is regenerated. The final stage in the regeneration is the redifferentiation and the pattern formation. So once the cone begins to elongate, one can usually identify cells that are beginning to redifferentiate. The cartilage cells forming around the end of the amputated bone are the first to appear, and later the other mature cell types appear. As redifferentiation proceeds, the rudiments of all the bones distal to the amputation level are laid down, initially as condensation of precartilage cells. By about 6 weeks after amputation, the entire limb pattern has been restored. Subsequent growth of the regenerate will make it almost indistinguishable from the original limb by about 3 months post amputation. So with this undifferentiated mass of cells will undergo redifferentiation, the functions being assigned to the cells. There will be the formation of cartilage, there will be the formation of bone, and then there occurs what is called as the pattern formation, that the digits has to be formed, and it will give rise to an organ which was lost, and it will be identical to that original organ. It's indistinguishable from the original which is there. So that one is formed, which is smaller, and then it grows to form the original shape and size. So you can see that there is a palate stage, there is a notch stage, and then there comes a digit stage, and the entire limb has been regenerated, which is there. So this is the mechanism how a limb is regenerated in salamander or in newt. Now, what are the factors that affect this regeneration? One of the factors is the creation of this regeneration blastema. So, this undifferentiated or dedifferentiated mass of cells. I wouldn't say undifferentiated, because undifferentiated is when you start with the cells, they are undifferentiated. But in this case, the undifferentiated cells differentiated, and then during the process of regeneration, they de-differentiate or lost their differentiation function. Therefore, we use the term de-differentiation. So, this creation of the regeneration blastema depends upon the formation of single mononucleated cells from the multinucleated muscle cells and epithelial cells. So, because they grow back to the, they go back to the progenitor cells, so this formation of the progenitor cells is very important, and a serum protein called as the thrombin plays a very important role in this. This was shown, for example, in here, that the de-differentiation of muscle cells, for example, in response to the serum, the newt myocytes do it, but the mouse myocytes do not. Mouse cannot regenerate a limb, but the newt can. And if it is seen that if they take the myotubes on a plate and they provide this serum containing thrombin, then they proliferate and they form mononucleated proliferating cells. But however, the mouse myotubes do not respond to the serum, and they remain as the multinucleated cell. So this was the experiment done by the Brockes, that the thrombin is the active serum component which can cause the formation of this single mononucleated cells in the newt. Then the creation of the regeneration blastema also depend on the maintenance of the ion currents driven through the stump. If this electric field is suppressed, the regeneration blastema fails to form. Another factor that affects regeneration is the proliferation of the regeneration blastema. So first was the formation of the regeneration blastema. Next is what are the factors that affect the proliferation or the growth of this regeneration blastema. It depends upon the presence of the nerves. So, this experiment will clearly show that there is a normal limb and there is a cut that has occurred. So, cut has been occurred and there is a regeneration which is there. When the cut has occurred, the nerve which is shown in the red line was intact. But in the denervated limb, if the limb was cut but there was no, but the nerve was removed, then there was no regeneration and it remained as a stump. However, if the limb was already aneurogenic, it did not contain nerve to start with, the limb still regenerated. So, it depends upon the presence of the nerve. If it was a normal limb, if it had already been an aneurogenic limb, then also it might regenerate. But the denervated limb, if the nerve was all there but it was removed, then it cannot amputate, then it cannot regenerate, the amputated limb cannot regenerate. There is a glial growth factor which plays a role here. It is needed for maintaining a high rate of cell proliferation. So, for the proliferation apart from the nerves, the glial growth factor is needed. The fibroblast growth factor 2, FGF2 is also needed, that serve as an angiogenic factor. It also promotes mitosis and patterning of the regenerating limbs which is there. Then it also depends upon the proliferation of the blastema also depends upon the mesenchymal factor FGF10. It is expressed in the mesenchyme of normal embryonic limb buds. The regeneration of limb is very similar to its original development. So, this mesenchymal factor plays a very important role in that. The next important factor is the pattern formation, the factors affecting pattern formation in regeneration blastema. The retinoic acid is one example. The gradient develops along the proximal distal axis and this activates genes differentially along the blastema. So, retinoic acid plays an important role in creating these gradient, and this gradient is important for the de-differentiation or redifferentiation of the de-differentiated blastema for the formation of the or regeneration of the limb. which is there. So we can look at the two-step model of amphibian limb generation. There is occurred an injury, then there is a wound closure which is there, and then after the wound closure there is a proliferation and the cell migration. So you know the apical epidermal cap has been formed. If it receives a signal, then only it will go on to form the regeneration. If there is however no signal, for example there is no serum or there is no thrombin which is there, then the scar will form. And the scar will remodel and the skin will regenerate. So the scar and the will regenerate, but the limb will not regenerate. Only the stump will be there with a new covering and new skin which has been regenerated. However, if it does receive a signal, for example the signal from the nerves, the proliferation and the de-differentiation will occur and the blastema will be formed. This blastema, if will not have any kind of signal, will just regress. But if it receives the signal from fibroblast and will receive the fibroblast growth factors, then the growth and the pattern formation will be there and the limb will be regenerated. So this is termed as the two-step model of the amphibian limb regeneration which is there, that how does this occur. Now we'll look at the regeneration in some other organisms. In some invertebrates and vertebrates. In invertebrates, usually there is a bidirectional regeneration, which basically means that if you cut an organism into two parts, both the parts will develop the remaining part. So so one organism cut into two will give rise to two organisms. So bidirectional regeneration. However, in case of vertebrates, for example, as we saw in case of salamander, usually it is a unidirectional regeneration where if you cut one part, only that part grows. That part will not grow the entire organism. So tail will not give rise to the newt, or the limb will not give rise to the cut limb will not give rise to the newt, but the newt will regenerate the limb. So this is a unidirectional regeneration. So we'll take the example of coelenterate Hydra now. Hydra belongs to the phylum Coelenterata or Cnidaria, and the name refers to the stinging cells, cnidocytes or nematocytes, the cells that are used to catch prey and eject toxins. It has a radially symmetrical body, the simple tissue organization. It's a diploblastic in nature. Only two germ layers are present, the ectoderm and the endoderm, that constitute the outer layer which is called as the epidermis and the inner layer which is called as the gastrodermis. And the two layers are separated by not a true layer but which is called as the mesoglia, or it's a kind of a jelly-like substance, and therefore it is a diploblastic animal. Important scientist, very significant role the Abraham Tremblay plays in the regeneration of Hydra. The Swiss scientist Abraham Tremblay began studying the freshwater polyp, now known as Hydra, in the 1740s. This work led to the remarkable discovery that Hydra could regenerate their heads and feet, and if cut into few pieces, all of them would regenerate to form new individuals. He was able to split the head of the polyp longitudinally and allowed the two heads to regenerate. By repeatedly splitting the new heads, he was able to generate a multi-headed animal, a monster-like animal that he named Hydra in reference to the mythological creature. Later, Linnaeus renamed the polyp Hydra in honor of Tremblay. Tremblay studied this creature in detail and performed numerous experiments, initially with the intent of determining whether the creature was a plant or an animal. He eventually became convinced, and correctly so, that it indeed was an animal. By studying Hydra, Tremblay was one of the first scientists to demonstrate that animals could reproduce asexually. So Abraham Tremblay has a very significant role in the study of Hydra. Now coming back to the regeneration part, when Hydra polyps are cut to pieces, they regenerate the missing structures completely. The Hydra's body can even be macerated into individual cells that after brief centrifugation form aggregates from which the heads and feet will appear over the course of few days, or the reconstitution of the entire organism can occur. The cells near the head of the Hydra top are likely to regenerate a head and tentacles. This polarity remains within the cells. The cells, if they were near the head region, then they will develop the head part, and if the cells were from the tail region, they can develop the tail part. So this is a kind of a polarity which is intrinsic in the cells, and they remain. The cells disaggregate and restack in various orders, shows regrowth according to their original position in the animal. For example, here the tissue from the head region surrounds that of the tail, resulting in tentacles on each end of the regenerated organism. The regeneration is not possible though from terminally specialized body parts such as the tentacles or the basal disc. So, from the entire body part the regeneration can occur, and depending upon the position in the original organism, that part can be regenerated. So, every portion of Hydra along its apical basal axis is potentially able to form a head and a foot. The polarity of the Hydra is coordinated by a series of morphogenetic gradients that permit the head to form only at one place and the basal disc to form only at the other. And evidence of these gradients was given by the scientist Ethel Brown, and his experiment is shown in the diagram, that when the hypostome tissue or the mouth tissue from one Hydra was transplanted into the middle of another, a new apical basal axis was formed and the hypostome started extending outward. In the B part, you can see that if a basal disc was taken and transplanted into the middle of the another, a new apical basal axis was formed and the opposite polarity was there. The basal disc started extending. And in the C part, if the tissue from both ends of one transplanted into middle of another, no new axis was there, or new axis has less polarity. So, there is a positional information which is there. The head region is giving rise to the head region, and the tail region is giving rise to the tail region. So, the cells, if they are transplanted to a different position, still they will give rise to from the part from where they are originally coming. This clearly indicated the presence of a head activation gradient which is highest at the hypostome, and a foot activation gradient which is highest at the basal disc. And 3 associated proteins have been found, termed as heady, head activator, and HIM301. Heady and head activator proteins are very critical for the head formation, and HIM301 usually regulate the number of tentacles formed. So, the gradient of these 3 proteins, they are highest at the head region and lower in the tail region. So, if the cell from the head region is taken and transplanted into the tail region, still it will give rise to the head region which is there. And this experiment again, tags those proteins and tells us about the these regional specification in the regeneration which is found. Then the graft of subhypostomal tissue, however, transplanted in the same region of the other, there is no secondary axis which is formed. Another experiment he did was that the graft of subhypostomal tissue transplanted in the same region of another decapitated host, the secondary axis was formed. So, if the intact head was there and the subhypostomal region was transplanted, there was nothing which was regenerated. But if it was transplanted onto a decapitated, from the one where the head was removed, the original axis was regained and a new head was also formed. If the graft of the subhypostomal tissue was transplanted into the lower region of another host, the secondary axis was formed. This clearly indicated the presence of a head inhibition gradient. So, the hypostome acts as organizer, it inhibits the formation of head in any other part of the body. So, when transplanted, it can induce host tissue to form a secondary body axis. It produces both the head activation and head inhibition signals. It is the only self-differentiating region. Head inhibition signal is to inhibit the formation of new organizing centers, and 3 active genes have been located in this head organizer: the Wnt protein, the apical end of the early bud, the goosecoid which is found in the hypostome region, and brachyury which is when hypostome comes in contact with the trunk. So, these 3 active genes gradient is present, and this hypostome acts as the organizer. We will talk in detail about this when we study about the embryonic induction which is there. So, you can see when the new thing is formed, then in the bud location the function of the head and foot inhibitor gradients are there, and the bud develops where the concentration of both head and foot inhibitors are minimal. So, at the top only head can develop, in the down only tail can develop, but where both the concentrations are minimal, a new bud can develop. So, this is a summary in regeneration in Hydra. The Hydra grows continuously and loses cell from its ends by budding. The head produces a gradient of inhibitor that prevents other heads forming. The head also specifies a gradient in positional values. When the head is removed, the local reduction in the concentration of inhibitor occurs and local increase in the positional value is there, and the formation of new head is there, however, without any new growth, thus giving an example for morpholexis. Thank you. So let's talk about the variation in the regenerative capacity in different organisms, and let's take up some examples of regeneration and particularly talk about the compensatory regeneration in the mammalian liver. So learning objectives for this session would be to understand this variation in the regenerative capacity in different organisms. We'll understand the basis of regeneration in some invertebrates such as Platyhelminthes, Planaria. We'll understand the regeneration ability in some other vertebrates and particularly the compensatory regeneration of the mammalian liver. We will also understand the absence of this regenerative ability as found in newts in mammals. Why mammals cannot regenerate the way the newts do. So let's look at the detailed mechanism. So this shows the variation in the regenerative capacity through phylogeny, ontogeny and aging. So there is a development which is shown, and then the adulthood, the young and the old is formed. You can see in Hydra and in Planaria there is a whole body limb regeneration that can occur. So we have already taken up the example of Hydra, which depicts the morpholexis regenerative mode. We will take up the Planaria whole body regeneration in this session. We have also talked about the urodale amphibians, in which the limb, the lens, heart, brain, spinal cord, jaws and hair cells can be regenerated. Then in anurans, the spinal cord, lens, head, brain and limb can be regenerated during the developmental stages. However, as they grow old, they might be, in old they cannot regenerate anything, but during young ages they might regenerate the digits. In fishes, the heart, brain, spinal cord, caudal fin and barbels can be regenerated, and caudal fin and pectoral fin usually for example in zebrafish and kelpfish can be regenerated during the developmental ages. In birds, the spinal cord can be regenerated during the development, and only the hair cells or in some cases the beaks can be regenerated during the young stage. In antlers, we'll talk about this. In deer, the antlers, the bone, the cartilage and the only regenerating organ of the mammals is this. Then in mouse, the heart and spinal cord has been found to be regenerated during the early developmental stages. The digits and hair gel cells still can be regenerated, but as they grow old, only this, the blood cells, the epidermal cells, the skin cells, the epithelia, endothelia, muscle and cells, bone and neurons are the only ones which can be regenerated. And same is found in case of human beings, that the heart and spinal cord can be regenerated during developmental stages, but as it grows from young to old, and in old probably even the blood, epithelia and these cells also cannot be regenerated. So you can note that the ability to regenerate the indicated system is present in most other in animal groups. For example, the lens, for example the neurons, and the lens regeneration is observed throughout the lifespan in newts, but it can only occur during a limited developmental window in axolotl, which is there. So this completely shows the variation in the regenerative capacity through the phylogeny, ontogeny and with the aging which is there. So let's understand the example of Platyhelminthes, Planaria. Planaria, as you can see, it's a freshwater flatworm. It lives under leaves and stones. It avoids light, that it exhibits negative phototoxic. It's a free living organism. It is not parasitic. It's a carnivore. It eats decaying meat, insects and larvae. It's 1 to 20 millimeter in size. It is a very long-lived organism, and it uses sexual or asexual reproduction. So there is a head, there is eye spots, there is auricle, which is the sensory organ, and there is a pharynx opening for food and waste on the underside, and there is a tail which is there. The planarians can regenerate a complete animal independently from the cut level and also from a fragments as small as 1/279 of their body. So we saw in Hydra that every cell has the potential to regenerate, but it retains the positional value. If it is coming near the head region, if the cell is coming from near the head region, it will develop the head parts. If it is coming from the tail region, it will develop the tail parts. But it will not give rise to the entirely new or the same organism which is there. But in Planaria, even any part can give rise to the entire Planaria, and in fact 279th part of the body can also give rise to the entire body. It might be smaller, but it will give rise to the entire body. So it combines both the processes of epimorphosis and morpholexis. The missing part is developed from the blastema, but the remaining part is reorganized on a diminished scale. So whatever part is missing It will give rise to, as we saw in the case of limb development in case of newts, but the part which is remaining from the original will repattern to form the entire organism, although it will be diminished in size. So, such is the regeneration found in case of Planaria. And the stem cells which are giving rise to this are termed as the neoblast cells. The 20 to 30% of all the cells in a planaria are neoblasts. Neoblasts divide by mitosis and can make more of themselves. They are the only dividing cells in the planaria. When a planaria is wounded, neoblast migrates to the site and begin dividing, as it is shown. There is a decapitation that has occurred. There is an epithelium and there is an underlying tissue. The epithelium will cover, so wound healing, wound covering will be there, and then regeneration initiation is there. There is a signal that is forwarded. Then the neoblast will reach there, and these neoblasts, so these are like stem cells. So these neoblasts will divide mitotically and will respond by giving rise to the functional, and the blastema formation is there. So there is a division. Neoblast can actually become any cell in the planaria that it needs, for example, the nerve cells, the flame cells, or the reproductive cells which is there. So that's how the regeneration occurs in case of planaria. So you can see that if the intact is cut into 3 pieces, then in 1.5 hours it shows like this. The middle part will give rise to both the anterior and the posterior part, and respectively all of them, the posterior will give rise to the anterior part and the anterior will give rise to the posterior part, giving rise to the entire organism which is found. So in Hydra, there is interstitial cells which play a role, and they can differentiate into the nerve cells, the gland cells which normally proliferate themselves, nematocytes and germ cells. The cells are actively dividing and can be easily killed by the drugs that inhibit cell division, for example, colchicine or hydrourea. The loss of the interstitial cells eventually results in epithelial hydra. The epithelial hydra can regenerate, but it will never develop the interstitial cell lineage again. However, in planaria, there are totipotent cells which are called as the neoblast cells, and they are wholly responsible for the regeneration, whereas epithelial cells do not proliferate. Example of tissue regeneration from a source different from that in normal ontogenesis. Neoblasts are derived from parenchyma that are of mesodermal in origin. They give rise to parenchyma in muscles, but also nervous system and pharynx that in normal development are produced by the ectoderm. So there is a difference between the interstitial cells and the neoblast cells. In neoblast cells, they are the true stem cells, they are the totipotent cells. They can give rise to any type of the cells. They themselves are coming from the mesenchymal cells, and they will give rise to the the parenchyma and the muscle cells only. But they have the potential to give rise to the nervous cells also, which in the embryonic stage was coming from the ectoderm. Interstitial cells, however, give rise to the epithelial cells, and they do give rise to the nerve cells and other cells, but if these cells are lost, then those cell lineages cannot be developed by any other cells. They are not stem cells as such. So these interstitial cells can give rise to the epithelial hydra if these are lost, which is there. So this was just to explain the difference between the interstitial cells and the neoblast, and also to understand a deeper mechanism that how the regeneration is occurring in case of Hydra as compared to the planaria which is there. Now let's look at the regenerative capacity of some other animals. For example, in annelids, the regenerative abilities are greater in the polychaetes and more primitive oligochaetes than in more specialized oligochaetes. The leeches are unable to regenerate. Most polychaetes and oligochaetes can regenerate a new tail when the body is severed. Ability is usually restricted to the anterior end, where lost segments are replaced either by the same number or by slightly fewer segments. The regeneration can even occur from a single segment, as it does in the polychaetes Ctenodrilus and Dodocearia. It involves several steps. The wound seals over, the blastema from beneath is formed, this surface is formed. The blastema probably arises from the differentiation of the existing cells, although mesodermal neoblast, which is a totipotent stem cells, migrate to the site of injury in polychaetes and many oligochaetes which is there. Regenerative structures are formed from this blastema. A hormone from the posterior part of the brain is essential for the posterior regeneration. This example shows the segment regeneration by lumbriculus, the 5 segment long posterior fragment in 0 to 21 days. So, you can see that the 5 original segments remain clearly delineated and distinct from all newly regenerated segments which are there. That is, each original segment is longer and more darkly pigmented than newly regenerated segments which is there, but that segments can be regenerated as shown in this annelid example lumbriculus. In mollusks, the regeneration ability is very poor. In arthropods, the regeneration is limited to formation of new appendages. For example, in crustaceans, it can occur at any stage of development. Crabs, lobsters, crayfish, shrimp and barnacles, there are around 40,000 species, 2 pairs of antennae. Unlike insects, crustaceans have appendages on their abdomen, and some pictures of the crustaceans are there, and they at any stage of development can probably regenerate their lost appendages. In insecta, the regeneration is restricted to larval stage. The regenerated limb does not reach the normal size. The steps involved include, after amputation, the wound is covered by the chitinous plug underneath, a regeneration bud formed, limb formation by epimorphosis, and limb is not apparent until the next molt. Several molts, limb reaches the full size. So, it is apparent only during the next molt, and after several molts the limb reaches the full size. So, only it is restricted to the limb regeneration, and it follows the mode of epimorphosis which is there. In echinoderms, the starfish, brittle stars and sea lilies, a complete new animal can grow from a small fragment such as arm. In some species, for example in Lynchia multiflora and Echinaster luzonicus, one of the arms will virtually pull itself away, regenerates and forms a new animal, as it is shown. One of the arm has developed and it will just pull itself away and probably will give rise to the entire new organism which is there. So, good regenerative ability is found in case of echinoderms. In fishes, the regeneration is again restricted. The fins can regenerate but not the tails. So, as it is shown, that is the after days post amputation, that is 0 days post amputation, and that is 10 days post amputation. So, the fins can develop but the tail fin cannot develop in regenerate in case of fishes. In amphibians, we have already seen the example of urodels, the newts and the salamanders. The limbs, tails, external gills, upper and lower jaws, parts of eyes such as lens and retina all can regenerate. In case of anurans, the frogs and toads, the regeneration ability is restricted to the larvae, and the limbs and tails are the only organs that can regenerate in the larval stages of frogs and toads. Coming to the reptiles, the lizards, the tails can, however the legs and digits cannot regenerate. The regenerates follows autonomy, the self-mutilation, a special adaptation which helps animal to escape being caught by a predator. So, it can leave its tail when a predator attacks, and then it can regenerate its tail again. The tails are broken off at a preformed level near the base of the tail. A regeneration bud is formed, giving rise to a new tail. The new tail, however, differs from the original. The vertebral column is a simplified one, and the scale covering the regenerated tails are different. Coming to the birds, the parts of beaks can be regenerated, or some hair cells can be regenerated, otherwise the regenerative abilities are poor. And last but not the least, regeneration in mammals. The regenerative ability is reduced to tissue regeneration or wound healing. So, it is basically up till the wound healing which is there, a repair mechanism which is there, and usually ends up in the formation of a scar. However, some of the tissues can regenerate completely, or what is termed as a compensatory regeneration, as we see in the case of mammalian liver. So, what happens in the liver is that the differentiated cells divide to recover structure and function of the injured organ. So, we know during the developmental process, when the cells have differentiated, they cease to grow, they cease to proliferate, they acquire a particular function and they start performing that function. So, initially they were progenitor cells, they were undifferentiated cells, and then they attain a specialized function. There is a morphogenesis that occurs, there is a differentiation that occurs, and they gain a specialized function and they cease to grow. The cells which still continue to grow and remain as progenitor cells are called as the stem cells. in the compensatory regeneration, the differentiated cells again start growing. So, the cell starts dividing to recover the structure and function of the lost part, which is, or the injured organ which is there. This is a standard assay for regeneration. For example, the liver regeneration, the standard assay is what is called as the partial hepatectomy. The specific lobes of the liver are removed, leaving the other lobes intact. So, the regeneration occurs by the cell division or the proliferation of the existing tissue. The regenerating liver cells do not de-differentiate. So, it is not that the differentiated liver cells will lose its function, go back to form the undifferentiated mass and then re-differentiate it to form the liver. It does not happen in that manner. How it happens is that the differentiated cells will retain the ability, will regain the ability to divide again, and they will take up the function of the lost part which is there. So, de-differentiated does not occur during the regenerated liver cells. There is no regeneration blastema that is formed. There are 5 types of liver cells which are there: the hepatocytes, the duct cells, the fat storing or the Ito cells, the endothelial cells and the Kupffer macrophages, and they divide and produce more of themselves which is there. So, this diagram depicts the liver regeneration, how this occurs. When there is a partial hepatectomy or there is a liver injury which is there, then there is an increase in the LPS, which is a lipopolysaccharide. There is an increase in the level of lipopolysaccharides, and through the portal blood flow, it goes to the Kupffer cells, and it is sensed by the Kupffer cells, and the Kupffer cells will release more of the cytokines, which is the IL beta, interleukin beta and tumor necrosis factor alpha, TNF alpha. So, it will release these 2 cytokines, and these 2 cytokines, in which TNF alpha also increases the production of IL beta, and both these cytokines will then act on the hepatocytes. Partial hepatectomy also increases the release of some metalloproteases, which might also influence the hepatocytes. Then there are stellate cells which releases the HGF, the growth factor, and TGF beta, which is also the TGF beta act as an inhibitory factor on the hepatocyte. However, the HGF will enhance the proliferation of the hepatocyte which is there. So, these hepatocytes are arrested at G0, G1 phase. The TGF beta will be inhibitory on the hepatocytes, and this will induce this G1 cycle, cell cycle. It will reinduce the cell cycle, and it will reinduce the hepatocyte to undergo this G1, to enter the S phase or the synthesis phase which is there. Apart from the proteins or hormones or the cytokines which are released by the injured organ, there are other hormones which also play a role, or the other molecules which play a role. For example, the thyroid will release T3, the pancreas will release insulin, the adrenal gland releases norepinephrine, and the duodenum releases epithermal growth factor EGF. And all these influences the injured hepatocyte to enter the G1S phase, and these hepatocyte will undergo mitosis and will cause the liver cells to regenerate. So, basically the differentiated hepatocytes will undergo the cell cycle, re-enter the cell cycle and undergo mitosis to produce more of the liver cells, and the liver cell regenerates, which is there. So, in addition, the mammalian liver also possesses a second line of regenerative ability, and this is that if hepatocytes are unable to regenerate liver cells within a certain amount of time, then there are certain cells which are called as the oval cells, and they divide to form the new hepatocytes. So, the first line of regenerative ability is that the hepatocytes, the differentiated hepatocytes will proliferate and will regenerate the liver cells, and which will take over the function. However, if they fail to do so, there are oval cells. Oval cells are the small progenitor cell population that can produce hepatocytes and the bile duct cells. They are kept in reserve and used after the hepatocytes have failed to regenerate, and this oval cells will reproduce or will produce more of hepatocytes, and the liver cell regeneration will occur. We will look at 2 more terms related to the regeneration. One is called as the super regeneration, to analyze the stimulus for regeneration. The damage is important and not necessarily the loss. The development of a superfluous number of organs or parts of the body as a result of regeneration is termed as super regeneration. So, in super regeneration, the stimulus for regeneration that the damage has occurred is important. The losing of the part is not important. So, the original part is also retained, but there is a damage or an injury, and therefore a new part will also develop. So, the development of a superfluous number of organs or parts of the body is termed as the super regeneration. Then there is another term which is used in context to regeneration, which is called as heteromorphosis. In heteromorphosis, a different organ develops from the one that has been removed. The concept of a regeneration territory comes here. If a part of the territory remains intact, the complete system may be restored. If the whole territory, however, is lost, it cannot be renewed at the expense of other parts of the body. So, it's important. So, heteromorphosis is when, for example, a limb is amputated, but some other part will grow up from there, because the entire territory or the region of the limb is removed, and therefore the limb is not able to regrow again. So, that kind of regeneration is termed as heteromorphosis. Coming almost to the end of it, talking about that how newts can do it, but why can't humans can regenerate the way they do. We tend to regard urodale regeneration as an exceptional attribute, since closely related species do not possess this ability. The critical hypothesis is that regeneration is a basic primordial attribute of metazoans, lost secondarily for reasons which are not really understood. And looking at the difference in the mechanism of regeneration, the de-differentiation in mammalian regeneration is absent, because there is an inadequate expression of genes, there is an insufficient intracellular signaling pathway which is there is irreversible expression of differentiation which is there, the structural characteristics that make de-differentiation impossible, or there is adaptive immunity, the cost of regeneration, that is it important that we regenerate the missing part or not. So, what will be the cost of this regeneration? So, probably because of these reasons that the de-differentiation in mammalian regeneration does not occur, and therefore it cannot develop missing or the lost part as the newts or salamanders can do. So, as I said, deer antlers are the only adult mammalian regenerating organ which is there, and if we look at deeper into how this deer antler regeneration occurs, then unlike horns which grow from the base, the deer antler growth center is at the blastema-like tip, where mesenchymal cells undergo a continuous process of cellular differentiation into abundant vascular tissue, cartilage and skin and bone. So, deer antlers can practically develop by itself, it can grow, regenerate properly by giving rise to the vascular tissue, cartilage and skin and bone, just like the newts do. So, there is a blastema-like tip which is there, and the growth occurs in that kind of a form. So, that is all about the regeneration and regeneration capacities and abilities in different invertebrates and vertebrates that we discussed.
Thank you, dear friends, for watching our show. Stay tuned and keep watching. Thank you.